Dear students, up till now we have discussed that DNA and RNA, they are macromolecules of nucleic acids, and they are composed of smaller subunits that are known as nucleotides. These are the subunits of DNA as well as RNA. And these subunits, they repeat themselves in a regular way or in a regular manner to give rise to large molecules of DNA and RNA. Now we'll see what is the composition of a nucleotide. Nucleotide consists of phosphate group, number 2, a 5 carbon sugar, or you can say a pentose sugar. Penta means 5, so pentose sugar will be the one having 5 carbons. And number 3, the nitrogenous base. Now all the things are attached to this central sugar, 5 carbon sugar or pentose sugar. How this is attached? Let me draw a diagram for you to show how these are attached. You can count the number of carbon atoms: 1, 2, 3, 4, 5. So this is 1, this is 2, 3, 4 and 5. Our point of concern is 1, 3 and 5. Now first of all, let us complete the valencies of all of the carbon atoms. So the nitrogenous base is attached to this carbon. Here, nitrogenous base is attached. Then phosphate group is attached to carbon number 5, and an OH group which is attached to carbon number 3 is also of very much importance, because this will help these two functional groups, the OH group present at carbon number 3 and the phosphate group which is present at carbon number 5. They are responsible for interlinking different nucleotides. Now this is a complete nucleotide. Another nucleotide, when it will come, its OH group will form a bond with the phosphate of this already existing nucleotide, and this bond will be known as phosphodiester bond. Phosphodiester. Let us suppose that there is OH of another nucleotide. So this bonding between phosphate of already existing nucleotide and the OH of the next nucleotide will be known as phosphodiester bond. When this bond is formed, they will eliminate a water molecule. So you can say that it is kind of dehydration reaction or dehydration synthesis. Because of joining of two nucleotides, because of this bonding, the 2 units polymer that results from this bonding will have, now the 5 end of this nucleotide will be free and the 3 end of this nucleotide is still free. So this will form bond with any other nucleotide here, and this 5 end will form the nucleotide phosphodiester bond here. So this chain will extend. This can extend throughout the whole length. So they can link to another nucleotide. In this way, many thousands of nucleotides, they can join together in long chains. So the linear strands of DNA or RNA, no matter how long, they will always have a 5 free end and a 3 free end. And this 5 to 3 chain is very important in the structure of DNA. Now we will see the specific bonding between the different nitrogenous bases, because this is a linear structure, but we have seen that DNA is always present in this form. So what is the phenomena that gives DNA this specific structure? Now we will discuss the specified bonding between the nitrogenous bases of the two strands of a DNA molecule. I hope you have understood nucleotide very well.